So in this final PowerPoint about vision, as I said, we were in the visual cortex, the first cortical processing of visual information, and we really still have things pulled apart into features, and we really want to put it back together so that we can recognize objects and shapes and things in our environment. And that's what we're going to do in these visual pathways past V1. So the striate cortex, or the primary visual cortex, communicates to the extrastriate cortex, and then we see that information gets split into two streams of visual analysis. So we're receiving signals from the lateral geniculate nucleus, passing those signals on to V2, as it is called, and it's going to have larger, more complex receptive fields, and then V2, or extrastriate cortex, is going to pass those signals along two pathways. One, going ventrally, so downwards in the brain through the temporal lobes, what we're going to call the what pathway, where we do our object recognition. And two, it's going to send information up dorsally, so up through the top to the top of the head to the parietal lobes, or what we're going to call the where pathway, or how we navigate through our world. I'm going to, at the very end, talk about it as maybe a how, so how we navigate through the world pathway. So I have some pictures of brains here that look kind of complicated, but I've tried to line them up with the words. And if it helps to go through this slide twice, I don't know. You might just go back and forth between the brains and the words. I've tried to line these up because I'm first going to talk about the dorsal stream, which is going up through to the posterior parietal cortex, so up to the parietal lobes for processing where information about where things are. And you can see that happening on the brain, going from primary visual cortex, V1, through V2, extrastriate cortex. We can see in there, pointed to in the green, MST and MT, which are the middle temporal area and the middle superior temporal area of the cortex, where we're going to process movement or motion that is important for figuring out where things are. We're going to send that information through those areas also for figuring out the form of we have to be getting information about the form of about motion in order to understand form. So it's important for the what area as well, but it's very important for where things are. So it's right in the middle of that stream, going up to starting in the posterior parietal cortex for figuring out where. And so those MT, MST are important in processing motion and figuring out where things are. And I have underneath that word akinetopsia. What this means is these are people who have damage to, it's usually the MT area, where we see that they are lacking the ability to perceive motion or movement in their environment. Quite dangerous. I'm going to talk a little bit about that on a later slide. And then as we move down to those second two brains, that we're getting that information into the inferior temporal areas, those temporal lobes that are the ventral stream, the what pathway. Okay, again, moving from V1, visual cortex, through the striate cortex. This time we're going through V4 very clearly, which is sometimes referred to as a color area. I don't love calling it that because we do process color in various regions of the brain, and it's quite complicated, and again in various regions. But this area, V4, I think I mentioned it in previous slides, where if we have damage to this area, this is where we see that cerebral achromatopsia, where they lose their color vision. And depending on the damage, they can lose it completely. They might lose color constancy. It really depends on the specific location and the extent of the damage, just how much they lose, or whether their world becomes really truly black and white. But V4 is clearly very important for color vision, which, as you know, and especially if you're like me and you really don't see very well, your color vision is very informative. So those stark differences in color, when I take off my glasses, red lights and green lights, I see them pretty well. McDonald's signs, I can see aspects of that because of the differences in color that give me enough information about form that I can figure out what I'm looking at. And then a couple more areas that are mentioned down there. So areas before IT, the infratemporal area that I'm going to talk about briefly with visual agnosia, and the fusiform face area, sometimes called the fusiform gyrus, sometimes called FFA. You can see that we're moving into the posterior inferior temporal for some of that information. As we get into the a little bit more anterior inferior temporal area where we are doing our complex shape analysis. So now we're talking about neurons that are their receptive fields. We're getting beyond feature detectors. Like I said, we really don't want to keep our world pulled apart into features. As we're moving towards that complex shape analysis, at somewhere in the temporal area, Tanaka et al. In 1991 and 1995, they found cells that are responding best to complex stimuli. I sometimes call this the lollipop neuron. I really don't think that that's what they called it, and I've never seen that in my textbooks, but you can see that in A, what's being shown at the bottom there is neural firing, this how much neural firing there is. You can see this peak of neural firing to what looks like a lollipop. If this was a monkey, if the monkey was just shown the stick of the lollipop, in B you can see there's really not any neural firing. If the monkey is shown just the top or the circle part of the lollipop, there's a tiny bit of neural firing, but really still not much at all. If we show the monkey something that is pretty lollipop-like, but has a square top instead of a circle, I don't know how well you can see it. There's neural firing, but it's not quite as strong as the neural firing for this neuron, this lollipop neuron that is responding best to what looks like a lollipop to me in A. And just so I say this before I continue to go through some of these areas of just that showing that this is a pathway that is helping us to recognize objects. The inferior temporal cortex contains cells now that are responding to specific complex shapes. We're going to see some areas that are modules for kind of categories of objects, but these cells are now insensitive to some of the distinctions that are critical to the cells that we've been describing earlier, because remember, in the all the way up to and through the visual cortex, we had this retinotopic map. So what we were responding to depended on the location in space. But now these neurons are going to give us the recognition of objects, which can now be moved in space, regardless of where this person's face is in my visual space. This is this neuron that is responding to this person's face. It's going to fire. So cells in this cortex are going to respond to familiar objects. So this, if I first draw your attention to the picture at the bottom left, that we are looking from the bottom side of the brain, and you can see, so it's flipped around so that we're pointing to the fusiform gyrus, which is down on that right side in humans, but we're down in that infratemporal cortex, the inferior part of the temporal cortex. I'm going to start by talking about visual agnosia, which is when people have damage to this pattern pathway somewhere in the temporal cortex, or depending on exactly where the damage is in the severity, they might lose certain objects. But if they're losing this area that is the pattern pathway in the temporal cortex, they have this inability to recognize objects, despite the fact that they have satisfactory vision. If you're interested in this kind of stuff at all, you might want to read The Man Who Mistook His Wife for a Hat by Dr. Oliver Sacks. It is a book where the neuropsychologist Oliver Sacks describes some of his cases as kind of case studies. He tells you about the specific brain damage, and he tells you a bit about the behavioral outcomes of what's happening with these people. And the one that the case study that the book is named for is The Man Who Mistook His Wife for a Hat, that they take him to Dr. Sacks because he's really not recognizing objects anymore. He runs into a parking meter and says, "Excuse me, ma'am," and he can describe things. So His vision is just fine as they're testing his vision. He can describe the features and what things look like, but he cannot put it back together to recognize those objects. And it is named for him leaving Oliver Sacks's office one time and going over to his wife and trying to pick up his wife's head as if it was his hat on the hat rack. And I'm going to distinguish this here from prosopagnosia, where again it depends on the location and the specificity, and the severity and where that damage is exactly. But for prosopagnosia, that damage is to the fusiform face gyrus, or the fusiform face area, sometimes called the fusiform gyrus, to that area specifically, that area of the inferior temporal cortex. Okay, I keep coming back to this idea that what the specific outcome, behavioral outcomes, or the specific perceptual disabilities, if you want to call them that, that we see are going to really depend on the specific location of the damage and the severity of the damage. So this is talking about a really interesting patient who shows a visual object agnosia, so an inability, so they're missing some of that pattern pathway, and some, I'm assuming actually it's part of the infratemporal cortex where they have categories of objects and not the pattern pathway, because their fusiform face area was maintained. They did not have damage to that. So this patient could recognize the face in this painting, or that there is a face in this painting, but could not recognize that it was made up of flowers and vegetables, and couldn't recognize any of those flowers and vegetables that make it up. And I'm going to talk for one more slide, I think, about neurons getting beyond feature detectors, because I've talked about the fusiform face area and I've showed a lollipop neuron, as I've called it, but we have these areas. We're definitely getting beyond lollipop neurons as we have these areas that are responding to categories of objects, really, or categories of things. So here we see the parahippocampal place area in A, and it is responding to, and you can see where it is. It's as it sounds around the hippocampus, parahippocampal, and it responds to the top row, so those places. And this is, you can notice, whether it's an outdoor scene, whether it's a building, whether that third one is like an empty corner of a room, and then the fourth one really looks to me like Legos that are maybe giving some indication that you can navigate through, that you would navigate through them somehow. So even just suggesting kind of navigation. Whereas if we're looking at any of those objects on the bottom, or those scrambles on the bottom, that parahippocampal place area is not responding in the same manner. And we also see the extrastriate body area that's being shown down in B. And these are fMRI images that are showing quite distinctly the regions of the brain that are responding, in this case, to parts of the body. And you can see it's any, like the arm, some body part, a whole person, a stick figure, a shadow figure. We're going to respond to the body. But as you can see down below, other objects not so much, scrambles not so much, and faces not so much, because we have a different area, the fusiform face area, that is specialized for faces. And as I've talked about these modules, which most of the textbooks do, I've stolen this. I think this is from Goldstein, because I really think it's important to keep in mind that even though we have these specialized areas that are activated, and we're seeing really maximal response in a certain area. So my fusiform face area, for example, for faces, I'm going to keep coming back to that one because it's easy to point out on the slides. We're seeing distributed activity over a wide region of the brain for any of these. So what the left is showing is the maximal response, as if you remember, we're doing that subtraction kind of logic, or as we're subtracting out when I'm responding to other objects, and I'm subtracting that out from faces. You can see that in that red in the bottom right there is that fusiform face area. I see the maximal response. We see some maximal response in other areas, but that's where it's strongest at the fusiform face area. But if we go over, so now if you go over to the brains that are showing just responses to houses, responses to faces, I'm going to pick the one for faces, and you can see how they usually do this is often is the yellow is the maximal highest activation, and as we're getting into reds really down to blues and greens, we're getting into lower and lower relative activation. As we're in the looking at faces, you can see that there is activity widely distributed across the brain. We just see a maximal activity at the fusiform face area, again on that bottom right area. So I'm going to get to talking about motion perception now, those middle temporal area, the middle temporal area, and the medial superior temporal area. I'm going to start with the MT, middle temporal area, or what's sometimes called V5. This is going to respond to, so neurons are going to respond to stimuli that are moving in a particular direction, and it's going to tell us about the direction and speed of movement. So people with akinetopsia, they are people who have damage to this area, to the middle, to MT and to V5. And we've learned that, we've learned about this from various patients who have this damage. One is patient LM. And I'm actually not remembering if I'm describing LM or another patient, but I have the stories of a woman who, something she cannot see is when she's pouring water or liquid into a glass, that movement of the liquid through time is actually telling her telling us when the glass is getting full. And so for her, it's just this kind of frozen, and then it's overflowing because there's no kind of movement that's telling her where she is and how full that glass is. It's also pretty dangerous for her to leave her house and try to walk around in the world without somebody else there, as how fast cars are going, exactly how far away they are is difficult. They'll be small and then they'll be big. So she doesn't have that kind of information about movement that can be really quite dangerous. I have this hyperlink here of form from motion that we are receiving information about objectness. As I said, it's really clear going up to the where pathway that pathway has to go through these middle temporal areas that are telling us about motion, because as things move, they are changing their where they are. But it's also important for figuring out objectness and form, which is shown in this video. And I think your author puts it in the medial right occipital lobe. And that is certainly true, but I have here with a question mark of the superior temporal sulcus. That superior temporal sulcus is where we are perceiving aspects about, so for my specific example, where we can see that these are people, and that this is a biological kind of motion that that is in the superior temporal sulcus, as looking at eye movements and mouth movements for trying to figure out aspects of our social world. In MST, and specifically in the dorsal part of this medial superior temporal cortex, our neurons are responding to expansion, contraction, or rotation of visual stimuli. And so what this is going to tell me is if I'm looking at the swing set and it expands, it gets larger, that's because I've moved toward it, right? And if it rotates over to the right like that, that's because I've tilted my head. So MST is really telling me about my own movement relative to the environment. And finally, we get back to the where pathway and the discussion of whether or not we might talk about it as a how pathway, as it is telling us about where things are in space, but it's also that is part of our interacting with our things in our environment. So it's actually Goodale and Milner and some of their colleagues who have worked with some people who have damage to the dorsal stream. The one I've read the most about is DF, as we usually use their initials to refer to them, but then also to keep their privacy safe. And I think that's who's being discussed in this first bullet point. I have a few examples from different classes and different textbooks of DF. So this woman with damage to her dorsal stream, she has no difficulty recognizing line drawings. So her wet pathway is intact and is not at all impaired, but she has trouble picking up objects. And so with working with this person, they could see that it appeared to be more of how to interact with those objects in space. So they want to call it a how pathway. For me, I'm not sure that I want to say it's a where pathway or a how pathway. Maybe I think it's really a where pathway and a how pathway, because those locations of objects in space, where things are, they must be, that is involved when we are trying to navigate through space and interact with objects in space and direct our movements towards those objects in space. And again, coming back to this idea of it's the location and severity of the damage. So sometimes it causes, so depending on the damage, we might see difficulty directing particular areas of the body to certain regions of space. So for example, a man with left parietal damage has particular difficulty with moving his right arm to go, his right hand to go pick up the cup, as that is the left parietal, that specific region of damage. Okay, and I do get to have a summary slide as we are finished with vision, which feels really good to me. I love vision, and I always go on a little bit too long. I think I actually cut out some things and did well, personally. But so we have to talk about the stimulus and the eye, how the eye is getting that information to the retina, the importance of the retina, all of the photoreceptors and the neurons that are then going to take, and then the retinal ganglion cells sending their axons, that optic nerve, to bring that information to the lateral geniculate nucleus and the superior colliculus. The lateral geniculate nucleus, the LGN, sending that information on to the visual cortex. And we have talked about in various places here, receptive fields, what neurons are responding to, as well as organization or mapping of those structures. And then we need to get beyond V1, right? We need to get to where we are recognizing objects, so we know what we are looking at, putting those features back together into faces and houses and places and bodies and animals and everything, as well as allowing us to navigate and interact with our environment. So where are those objects? How do I interact with them? And so if you have questions about any of these aspects of vision, it's a good place to go back and see where your questions are so we can finish this on Monday.